Protein-producing synthetic cells offer a versatile platform for studying the origins of life, as well as synthetic cell-based therapies for advanced drug delivery. This method is simple and affordable for RNA and protein expression, and can be applied for on-site therapeutic protein production directly inside the body.
These systems can be used for treating a wide range of diseases, from metabolic diseases and protein replacements to cancer and possibly diabetes.
This multi-step protocol has defined stopping points. When performing it for the first time, it is recommended to divide the work over several days.
The protocol for synthetic cell production starts with the preparation of the cell-free S30T7 lysate. Next, the membrane lipids and cellular nutrients solution are prepared to simulate the inner environment and to support protein production. The final step is the preparation of the synthetic cells themselves. Prepare duplicate starter cultures in 100 milliliter Erlenmeyer flasks. To each flask, add 5 milliliters of LB media, supplemented with 50 micrograms per milliliter of ampicillin, and inoculate with a single E. coli colony. Grow the culture overnight in a floor incubator shaker at 250 RPM and 37 degrees Celsius. Next, prepare 2-liter baffled Erlenmeyer flasks containing 500 milliliters of TB media supplemented with 50 micrograms per milliliter of ampicillin. Inoculate each 2-liter flask with a 5 milliliter starter culture. Place the flasks in a shaker at 250 RPM and 37 degrees Celsius. Monitor the cultures.